the development of transgenic banana, hoping to get an improved variety of banana that can tolerate a disease called fusarium wilt. So this fusarium wilt is a fungal disease that is obviously caused by the fungal, which name is Fusarium oxysporum forma specialis cubensi. During that time, so basically when I entered UPM as a tutor, I was given a suggestion by the head of the department during that time, Prof. Raha, to go to the UM to be interviewed by this one professor, Professor Nurjulani. She during that time got a grant working on this transgenic plant, which is banana. So I did not choose the plant, but rather I was given the task by my supervisor. But after I started doing my PhD, lab work and all those stuff, then I started to really appreciate this plant. One of the reasons, this might not be related to science, but I accidentally found out in the Quran that banana is one of the plants that were mentioned in the Quran. So I think that is more meaningful for my research now. So knowing, I assume that you have some basic knowledge in GM production. So as a background, Fusarium wilt is one of the most threatening fungal diseases that affect banana plantations across the globe. In Malaysia at the moment might not be as bad as it used to be in the past, because now we have more recent disease that is caused by bacteria, but it used to be one of the most threatening fungal diseases in Malaysia. But currently across the globe it's still a serious disease, this Fusarium wilt. In order for us to develop a transgenic plant, in this case transgenic banana, that can tolerate this disease, first we have to, of course, identify the gene that we are interested in lah. Okay, so in my previous research group in the UM, there was a student, a master student, that already did microarray study. You know microarray kan? Okay, microarray study to compare the differential gene expression of tolerant and susceptible banana. So during that time, there is a variety called mutiara. So mutiara banana, and it was said that mutiara is tolerant against fusarium. So this particular master student, he did a microarray study to compare the gene expression of mutiara, and I'm not sure what is the other the variety that he used during that time. But upon getting the results of the microarray study, he noticed, or he analyzed the genes that were upregulated after the fusarium infection. Then my supervisor picked a few genes to be given to a few students that just entered the lab, and during that time I was one of them. So I was given a gene that is called PR10, pathogenesis related 10. So PR10, if you read literature, PR10 should possess antifungal property. So it is said that PR10, when they studied PR10 in other plant species, not banana, they said that PR10 should have possessed antifungal property. So I think this make sense, because I am fighting against fusarium wilt, which is also caused by fungus. So if I aim to overexpress this PR10 in banana, hoping that the new variety or the new clone that I developed would also have improved tolerance against fusarium wilt, since I overexpressed the genes that should have antifungal property. So when I got the genes, it's actually a very short fragment, because microarray you won't get a full length of the genes. So from that short fragment of PR10, I did an experiment called RACE, rapid amplification of cDNA ends, to get the full length of this PR10. Then I analyzed the sequences and only clone the coding sequence of this PR10 into into Picambia vector and transform into Agrobacterium. So I co-cultivate this Agrobacterium that already had this PR10 with the banana cell suspension of, during that time I used Brangan, pisang Brangan. So basically this PR10, the sequence originally came from Mutiara banana, Mutiara banana, but when I worked with PR10, oh, I forgot to mention that I amplified this PR10 from wild banana, not Brangan. Wild banana is called Malakensis. So pisang yang you selalu jumpa kat hutan-hutan, pisang yang from hutan lah. So I amplify this PR10 from wild banana, but I overexpress back in Brangan. So basically there are 3 different varieties involved in this aspect. Using tissue culture technique, you have to culture this suspension culture that should have your PR10 inside it. So I use antibiotic selection, just like your lab lah. So I have to select for the clones that, for the bacterial clones that contain my gene of interest using antibiotic selection and also PCR. So I amplify PR10 genes from the selected clones. Then after that, using these few analyses, I regenerate the plants, the banana plants, these Brangan plants, putatively harboring this PR10 gene. They should overexpress. So how? The next stage is to verify whether these so-called clones that I regenerated tolerate or can tolerate fusarium milk. So basically, when you did tissue culture, you only have the plantlets inside the tissue culture bottles, right? So I took these plantlets that already were confirmed to have this PR10, verified using PCR and also qPCR. Then I planted them in the soil and acclimatized for 2 months. After that, I prepared the spore suspension of fusarium. So this spore suspension must be in a certain range of concentration, because we know that only a certain range of concentration can cause infection against or towards the plants. So basically what I did, I have this spore suspension with the concentration that I wanted. Then I uproot the plants. So for example, this is the banana plants that I grew from the tissue culture plant, tissue culture stage. So I uproot the banana plants. What I did, I just dipped the roots of the banana plants, the transgenic banana plants, inside the spore suspension culture for 2 hours. Then replant back in the soil. Then for the next 5 weeks, I need to observe the symptoms that they develop, because for fusarium milk, the infection started via the roots. So when the infection started via the roots, that's why we need to dip the roots in the suspension culture so that we can induce the infection. Then why we need 5 weeks to observe, because within these 5 weeks we can start to see the symptoms that develop on the leaves and also on the stems of the plants. So some of the symptoms you can see internal and external symptoms. For example, on the leaf of the banana plants, you will see yellowing and wilting symptoms on the leaves. And if you uproot the plants and you cut the pseudostem, you might be able, or actually you have to see the discoloration in the rhizome and pseudostem. If the infection advances fast enough, then the discoloration might already go up to the pseudostem. Otherwise, it might just linger around at the rhizome. So that indicates that the fungus already attacked the banana and colonized the xylem tissue. Because what this fusarium did to the xylem tissues is, when they produce the hyphae, they will colonize the xylem tissues, and you know that when the xylem tissues is colonized, then you cannot transport the nutrients and water from the roots to the upper parts of the plants, right? So that's why you started to see the wilting and yellowing symptoms on the leaves. So that's how the disease progressed. So the outcome of my research, I did not see any resistant clones, which means that among all the plants, the transgenic plants that I tested, there is none that is really resistant against the Fusarium infection. However, I did see some delayed symptoms when I compared. When you did experiment, you have to have your own control. So my control is the berangan that was dipped in just distilled water. So when I compared my transgenic plants with the control first, but berangan plants that were dipped in the distilled water. Second, berangan plants, not the transformed one, not the transgenic ones, that were dipped in also Fusarium. So comparing the berangan plants that are not transformed, that are not transgenic, non-transgenic ones, with the transgenic ones, the transgenic ones showed delayed symptoms. When I started to see the symptoms, it's already about 1 week after the non-transgenic ones. So there is no resistant clones, but I only managed to see some delayed symptoms among the transgenic. The easiest or the simplest one, contamination, tissue culture obviously, but one of the most challenging stage in my work is to get the full length sequence of PR10. I did mention about race, briefly about race. So I think I took about 1 year just to get the full length sequence of PR10. But eventually we did not clone the full length, we only chose the coding sequence. So the race itself took about 1 year. Then the transformation part, I think was quite okay, but the verification part was quite challenging, because when we work with tissue culture, we have to have a system to indicate how do you define line, because I use cell suspension culture. Cell suspension culture, to put it briefly, is a single cell. So starting from single cell to get a whole plant took me, I think, close to 2 years. Because okay, you have single cell, so cells need to keep on dividing, and you got tissues. Before that, callus, shoots. So from single tissue to the whole plant, I think they are close to 2 years. So that is one of the biggest challenges that I have during my PhD work. So it's more on time lah. Failure in the experiment is normal, but time is something else. And in 2011, I think I finished my lab work in 2000, maybe 2015, 2016.
4 years.
4 years, about 4 years. But I took some more to write thesis, waiting for the viva. But the whole lab work, I think 4 years plus. I think many people have bad perception about genetic engineering. We cannot really blame them, because they don't have the knowledge that we ourselves only have very tiny knowledge on genetic engineering. But compared to them, they might not have any knowledge at all, or even if they thought that they have knowledge, maybe readings from internet, but through internet readings, I think most of the information are already mixed and misled, right? So as much as we think that genetic engineering is good and could be beneficial to us, we must also play our role as scientists, and I assume you all are plant scientists as well, to convince other people. The closest one could be your family, friends. Just educate them, share some information on genetic engineering, so that they won't have any bad perception on genetic engineering. Because in order for us to produce a genetically engineered plant or organism, it really took us long time, and it's not only constrained to the lab stage. When you go to the real field, you have to get approval from government, from the people living in that area, For example, in Malaysia, we don't have that many field trials on transgenic plants because the regulations for us to do transgenic testing or transgenic plants testing in the real field is very difficult. Not really difficult lah.
Too much proceeding.
If I said difficult kan susah pula kan. But it's not that easy lah. If you just work at the lab stage, it's okay. But in order for this product to be released to the market, it still has a long way to go. So when you see a GM product on the market, just bear in mind that if that product already follow the correct regulations and procedure before they were released to the market, then it should be safe enough for the consumers to eat. So I think that is one of the misleading statements that misleading information that most people have. Maybe they thought that it's very easy to produce transgenic plants or transgenic organisms that could harm other people, but it's not that easy actually.